Hello everyone, this is Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be explaining you amino acids, chemistry and classification. Let's get into the details. Now you need to know every amino acid has got alpha carbon in the center. So I'm going to draw that for you. So here is the alpha carbon which is in the center, and as you know, carbon atom has got 4 valency. And these 4 valencies satisfied by, so amino group is one of the valency attached to amino group, and then this another valency attached by carboxylic acid group, and then hydrogen atom, and then we have R group which is also called as side chain. So these are the 4 groups that are attached to the alpha carbon. So this is the alpha carbon which is a central carbon in an amino acid. So every amino acid, it has got this arrangement. Only a difference between each amino acid is this R group here is the one that changes in every amino acid. That makes amino acids different. So how many amino acids we have? So in a human body, whatever the protein that you take, it has got a 20 standard amino acids or 20 primary amino acids. So every protein, it is made up of 20 amino acids. Some of the proteins may not have all 20 amino acids, but these are the 20 amino acids which are basically referred as building blocks of all proteins. So amino acids are building blocks of proteins. We have only 20 primary amino acids. What is the meaning of primary amino acid? A primary amino acid which is also referred as a standard amino acid. So it has got a genetic code, it has got a codon. Every primary amino acid or the standard amino acid has got a genetic codon, okay, and it will be part of a protein molecule. That is what we refer it as a standard amino acid. So I have already explained you the general structure of an amino acid. To repeat one more time, amino acid has got alpha carbon in the center. Then we have a carboxylic acid group attached, then we have amino group attached, and then we have H attached, and then the R group attached, okay, and this is how amino acid looks like. All right, so now based on the groups that are attached to the alpha carbon of an amino acid, basically I am talking about this R group here. So look at the R group, and every amino acid, all like 20 standard amino acids, out of 20 standard amino acid, 19 of them, the alpha carbon is it is basically asymmetric carbon or a chiral carbon. That means, so alpha carbon is attached by different atoms. Okay, let me explain that to you by taking example of glycine and alanine. These are the 2 simple amino acid. I am going to write down the structure of glycine. I am writing alpha carbon, then H, COOH, that's a primary carboxyl group, NH2, primary amino group, and then H. This is a glycine amino acid. Simplest amino acid in our body is glycine. Now I am going to write alanine structure. Alpha carbon in the center, COOH, that's a primary carboxyl group there, and primary amino group is NH2, H, and in glycine the side chain is CH3. Okay, now the difference here is in the alanine side chain is CH3. So this is alanine. So now the difference between glycine and alanine is only the R groups. I am circling now, R groups. Just pay attention to the R group. R group in glycine is just H, whereas in alanine is CH3, methyl group. Now look at glycine. So alpha carbon is attached with the carboxylic group, it is attached with amino group, and then there are 2 H. So that means the 2 similar atoms are attached to the alpha carbon, and that is symmetric. It's not a chiral or asymmetric. So that's why glycine is achiral. So that means it is symmetrical, right? Whereas alanine, look at alanine, R group is different. Then we have H, then we have carboxyl group and amino group. So 4 different groups are attached to the alpha carbon. So this type of amino acids, the alpha carbon of this amino acid is asymmetric carbon or a chiral carbon. Remember that. So that means out of 20 standard amino acid, 19 of them have such arrangement. Okay, so alpha carbon is attached with different groups. Okay, so now amino acids can be classified depending on their rotation in the light, polarized light in the solution, into L-amino acid, which is a levorotatory, and D-amino acid, which is a dextrorotatory. So nearly all amino acids occurring in a mammalian proteins are of L form. Make sure you remember this. This is an important point. Human proteins, nearly all amino acids that are found in human or mammalian proteins, they are of L form, levorotatory form. Okay. So now each amino acid except proline has primary amino group and primary carboxyl group. Let's look into that. So as I told you, the general arrangement of amino acid, it has got alpha carbon in the center. It has got primary carboxylic group that is attached. It has primary amino group that is attached. So carboxyl group and amino group that is attached to the alpha carbon. Now the primary carboxyl group or primary amino group, it is basically the carboxyl group that is attached to the alpha carbon and the amino group attached to the alpha carbon. We simply refer them as primary carboxyl group and primary amino group. When the amino acid is in individual form, they should not be interacting with the side chain. Okay, if it interacts with the side chain, we cannot call that as a primary amino group or primary carboxyl group. So that means out of 20 amino acid, 19 amino acids, whenever they are in individual form, their primary carboxyl group and primary amino group, it does not interact with the side chain. Okay, only one amino acid in our body, in a protein, whenever it is in free form, so its side chain is making an interaction with the amino group attached to the alpha carbon, and such amino group is called as secondary amino group. So that's the difference here. So every amino acid in our body except proline, it has got a primary amino group, whereas proline has got secondary amino group. That's an important point that you should remember. Alright, now we have seen the carboxyl group and amino group. Let me write down one more time. Here is the alpha carbon in the center. Then we have a primary carboxyl group, and we have a primary amino group NH2, then we have H, and then we have R group. That's a side chain. Okay. Now see this primary carboxyl group and primary amino group, whenever it is in individual form, and depending on the pH of the media, so these groups can undergo protonation and deprotonation, and that depends on the dissociation constant of these amino acids. Now the dissociation constant it is expressed as pKa value. So that's what I have written here. The strengths of weak acids are expressed as their pKa. pKa is a dissociation constant. Now the dissociation of weak acid groups depending on the pH of their surrounding media. Okay. So first we need to know in order to understand whether carboxyl group undergoes protonation or deprotonation. You know, protonation means addition of protons to a group. Deprotonation means release of protons from a group into the media. Okay. Now the average pKa of primary carboxyl group of amino acid is around 2.0. Okay, approximately 2.0. Whereas the primary amino group pKa is 9.5. pKa is 9.5. Now, when we take a physiological pH, that is 7.4. So let's take pH, physiological pH is 7.4, and let's compare pH 7.4 with pKa of amino group and pKa of carboxyl group. So in order to understand whether the groups will undergo protonation or deprotonation, we really need to know the relationship between pH and pKa. What is the relationship between pH and pKa? Now, you got to remember these three statements here. What is that? Number one, whenever pH of the media is less than the pKa, when pH of the media less than pKa, pH less means it is relatively having more protons. You know, pH is a negative logarithm of hydrogen ion concentration. When pH is less than pKa, the group will undergo protonation. Remember that. Whenever pH is statement number 2, whenever pH is more than pKa, the group will undergo deprotonation. That means it's going to release proton into the solution. pH more means it's a negative logarithm of hydrogen ion concentration, means proton concentration is less in the solution. So whenever third statement is pH is equals to pKa, that means whenever pH is equals to pK, at that time 50% of the groups will undergo protonation and 50% of the groups will undergo deprotonation. Let's understand that with this example. pKa of carboxyl group is 2, pKa of amino group is 9.5. Let's compare that with pH 7.4. So let's look at carboxyl group first. pKa is 2, pH is 7.4. So let's see that relationship between pH and pK of carboxyl group. I'll write pKa COOH carboxyl group. So pH is 7.4, pKa is 2. So that means pH is more than pKa. pH is more than pKa. When pH is more than pKa, that is statement number 2 here. Statement number 2, pH is more than pKa. What will happen to the group? Group will undergo deprotonation. That means this H here, COOH, that COOH, it is undergoing deprotonation. It is released into the solution and that becomes COO minus. Okay, whenever amino acid is in individual form in the blood under physiological pH 7.4, primary carboxyl group undergo deprotonation. It becomes COO minus and H plus is released. Now let's look at primary amino group. Now the pK of primary amino group is 9.5, pH is 7.4, so that means pH is less than pKa. Now let's look at statement number 1. Statement number 1 is pH less than pKa, the group will undergo protonation. So what happens here? Amino group, that is NH2 here, will undergo protonation. That means the proton that is released here into the solution, it can be used to protonate this NH2 and that becomes NH3 plus. So primary amino group is protonated because its pKa is 9.5, pH is 7.4. So that's why based on these statements we can understand whether the groups will undergo protonation and deprotonation. Okay, now at physiological pH 7.4, so amino acids predominantly, especially primary amino carboxyl group and primary amino group. Primary carboxyl group is predominantly deprotonated. Primary amino group is predominantly protonated. Then side chain, it depends. If the side chain has got any dissociable groups, depending on the pK of side chain, so it can undergo protonation or deprotonation. So we'll look into that when we look at a specific amino acids which have got dissociable group. Okay, so now when pH equals to pKa, at that time 50% of the group is protonated, 50% of the group will exist as deprotonated form. If it is, if just as an example, if pH is 2 and pK is also 2, right, for carboxyl group, COOH, right, 50% of the carboxyl group is as COOH and 50% will be COO minus. That's what it is. Whereas in physiological pH is 7.4, so generally this doesn't happen. pH never goes to 2. If that happens, the person will die. Okay, so it is just a hypothetical thing. So that's little bit about relationship between pH and pK. So now whenever amino acid has got equal number of positive charge and equal number of negative charge, so that exists as an ampholyte. Ampholites are having equal number of positive and equal number of negative charges. Okay, so here it is in the figure as it is shown here. In the figure you have alpha carbon in the center and amino acid primary carboxyl group, as you see here, it is deprotonated predominantly, COO minus. Primary amino group, as you see here, it is protonated, NH3 plus, and we haven't talked about R group protonation, deprotonation. We will see that later. Okay, so now let's look at something called as Zwitterion. What is Zwitterion? Zwitterion, it is a molecule which has got equal number of positive and equal number of negative charge, and net charge of a molecule is 0, and that type of molecule we call it as Zwitterion. So majority of our amino acids, they exist in Zwitterion form at pH 7.4, that's the physiological pH. Very few amino acids which we are going to see, acidic amino acids and basic amino acid in that category. So they will have either net positive and net negative or net negative charges. Other than that, predominantly most of our amino acids in the physiological pH, they exist as Zwitterion form. Now, let's look at another concept here called isoelectric pH or PI or isoelectric point. So, isoelectric pH is the pH at which the molecule exists in a Zwitterion form. Okay, so that means it will have a equal number of positive and equal number of negative charges at isoelectric pH or PI. Okay, so that's what is shown here. As you see here in this particular figure, let me show you. Zwitterion form, dipolar form, whenever pH is equals to PI or isoelectric point, you see there is one positive charge, one negative charge, one positive charge, so net charge is 0. But whenever pH is less than PI, as it is shown in the left side here, look at this. pH is less than PI. pH less means what? More protons are there in the solution. So when the pH is less than isoelectric point, at that time protonation will go on. Protonation, because pH less means more protons are there in the solution, it is going to protonate some of the molecules. So that means there will be protonation of carboxyl group into COOH. COO minus becomes COOH, protonation. So let's look at the overall charge of this molecule. Whenever pH is less than PI, look at the overall charge. It has one positive charge. That means molecule exists as a positively charged ion, a cationic form. Cationic form means it has a more positive charge there. Okay, so now let's look at the other side, right side of it. So whenever pH is more than PI, whenever pH is more than isoelectric point, at that time what happens? pH more means what? Less hydrogen ion concentration. That means there will be more deprotonation of molecules. That means this NH3 plus, it is deprotonated into NH2, and again, like the carboxyl group is already deprotonated. So overall it has net negative charge, one negative charge is there, so that is a anionic form. So that means whenever pH is less than PI, molecule exists as cationic form or a positive form, positively charged. Okay, whenever a pH is more than PI, molecule exists as anionic form or the negative charge, overall net negative charge. Remember that point. Alright, so that's what the statements I have written here. So isoelectric point for simple amino, before I make a statement, let me explain you how to calculate isoelectric point. So if example I will take here, I will write a simple example for explaining isoelectric point. I will take alanine as an example. Okay, so we have COO minus primary carboxyl group, and then primary amino group NH3 plus there, and then we have CH3. So basically we have a primary carboxyl group, primary amino group. These are the two dissociable groups. Okay, so you will have to take a pKa values of primary carboxyl group here, that is 2.0. pKa value of primary amino group, that's pKa of primary amino group is 9.5. So how do you find out PI, isoelectric point? So in order to find out isoelectric point, you need to take the average pKa of ionizable groups in a molecule. So what are the ionizable group in the in amino acid alanine? So ionizable group is primary carboxyl group and amino group. So carboxyl group is 2.0 and amino group is 9.5. So 2.4 plus 9.5. So that will be and then you divide that by 2, because we'll have to take average. So it will be 11.5 divided by 2. Okay, or if you just take, instead of just for the simplicity, if I instead of 9.5, if I write it as 10 here, so 10 plus 2, that is 12. Okay, so I'll just write it as 12 there. 12 divided by 2 is 6. So 6 is the PI. So basically it's an average of two ionizable group, and that's how you make you are going to find out isoelectric point for simple amino acids which have got two different ionizable group. So if an amino acid has got isoionic species or polyprotionic species or something, at that time you will have to take at the take the average pKa of isoionic species. Example, let me explain that to you. So instead of alanine, now I am going to write aspartate, amino acid aspartate. It's an acidic amino acid. I am going to explain that to you. So it is CH2 and COO minus. Okay, now look at ionizable groups here. So there is a carboxyl group attached to the alpha carbon, side chain carboxyl group is there, and the amino group here. Now these two so there are two carboxyl group and one amino group. So that means two carboxyl groups are similar here. So these are isoionic species. So how do you find out pKa value of this type of amino acid? So whenever in similar groups are there in an amino acid, you need to take the average pKa of similar groups. Okay. So for that means you will have to take average pKa of these two carboxyl groups. So for primary carboxyl group, pKa is 2. For side chain carboxyl group here, I will just write it as it's not an accurate number here, I just write 2 there, pKa 2 again. So what is the PA isoelectric point of aspartate here? So we'll have to take isoionic species, so that is 2 plus 2 divided by 2, that will be 4 divided by 2, that will be 2. So that is what is PI, isoelectric point of aspartate. So this is how you are going to calculate, or you are going to know how to find out isoelectric point of an amino acid. So if for simple amino acid, you take two ionizable groups and take the average of it. For polyionic species, polyprotic species, where similar groups are there, you take the pKa value of similar groups and take average of it. Okay, so that's about it. Now the relationship between pH and PI, I already explained you in the previous slide. So when pH less than PI, so there will be protonation going on, so it will exist as positive charged, and when pH is more than PI, so molecule exists as a negatively charged anionic form, cationic form and anionic form. We have seen that already. Now let's look at classification of amino acids. Amino acids can be classified in a different way based on the polarity of an amino acid, of variable side chain, polarity of R groups. You can classify an amino acid based on the variable side chain characteristics, that is the R group characteristic. We can classify amino acid based on the metabolic fate. We can classify amino acids, and also based on the nutritional requirement we can classify amino acids. So basically we are looking at 4 classifications of amino acids, number 1, 2, 3 and 4. Let's look into each one of them. So our very first classification of amino acid is a classification based on the side chain characteristics. That is, some of the amino acids have got a non-polar side chain, which are referred as hydrophobic because they don't interact with water. So the side chains of some of these amino acids, they don't make hydrogen bond with the water, and that means they are not soluble in the water. Whenever they are in the attached to a polypeptide chain, when they are part of a protein molecule, so they don't interact with water. So that's why all these are referred as hydrophobic amino acids. And majority of hydrophobic amino acids in a globular protein, if a protein has got globular shape present in the blood or cytoplasm, extracellular matrix, all these hydrophobic amino acids are buried inside the globular protein, while the hydrophilic amino acids, which will interact with water, right, so they will be outside. And if it is a membrane protein, you know membrane is hydrophobic. So if it is a transmembrane protein, let me write down the membrane. So consider this is the lipid bilayer membrane, and then if it is so I am writing a transmembrane protein passing through this membrane here, this is the protein. Okay, so you can divide that into number 1, number 2 and number 3. So it is divided into 3 parts. Number 1, it is on the extracellular matrix, and then number 2, it is passing through the membrane, so it is a transmembrane protein, and number 3 is in cytoplasm. Okay, now in the extracellular matrix and cytoplasm, water is there, so that means amino acids which are there in that particular portion, so they should be hydrophilic amino acid, whereas the one that is passing through the membrane, transmembrane protein, should have hydrophobic amino acid. That's how the arrangements will be going on. So what are all these hydrophobic amino acids? Here is the list. So alanine is an hydrophobic amino acid because the side chain of alanine is CH3 methyl group, and then valine, leucine, isoleucine. These 3 are hydrophobic amino acid because they have got this branched side chain, and you see they are all ending as methyl groups. Okay, within the branch, if you observe carefully, they end as methyl group. So there are 3 branched chain amino acids there. And then we have methionine. Methionine has got this thioether linkage. There is sulfur there which is not free and says methyl group, that's hydrophobic. And then tryptophan. Tryptophan has got indole ring. So I am basically circling the group there. This is indole ring. This is indole ring here, which is hydrophobic there. Phenylalanine. Phenylalanine, look at the side chain. It has got a benzene ring. Benzene. Phenylalanine has benzene ring, which is hydrophobic. And then proline. You know, proline has got the side chain which is interacting with the primary amino group. So in fact, proline don't have a primary amino group because it is not free. So it is quite different than any other amino acid. Let me just recap that particular point. Here is the side chain of proline. It has CH2, CH2, CH2. Look at this CH2. It has interacted with the NH3 plus, which is attached to the alpha carbon, making a 5-membered ring. Unlike all amino acids that you are seeing, primary amino group, it is free here. Whenever they are in individual form, it's all free. Whereas this primary amino group, it is not free. It is interacted with the side chain. That's why this amino group present in proline is referred as secondary amino group. That's why in order to differentiate this with other amino acid, it is sometimes referred as amino acid. Proline is referred as amino acid. So this entire group here, 5-membered ring, is referred as pyrrolidine. Pyrrolidine ring. That's what is the special group present in proline. Alright, so these are all hydrophobic amino acid and you must remember all these. So that is alanine, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine and proline. How do you remember this? The mnemonic is PIM, PAL, TV. You can remember like that. P is for phenylalanine or you can put it as proline also. I for isoleucine, M for methionine, another P. So let's put it as proline here because other P was phenylalanine. A for alanine, L for leucine, T for tryptophan and V for valine. So this is how you can remember as PIM, PAL, TV. Alright, so that's about hydrophobic amino acids. Now let me explain you a polar uncharged amino acid. That means these amino acids, they are polar in nature. They can interact with water. These are hydrophilic amino acid, but they don't have any net charge under physiological pH. What are those polar uncharged amino acid? We have glycine, which has side chain H. So it is soluble there. Serine has a hydroxyl group CH2OH, which is polar. It can interact with water. Threonine has got hydroxyl group in the side chain. And cysteine has a thiol group here, right, which can interact with another thiol group. Tyrosine has got phenol ring, phenol, it has hydroxyl group there. It can interact with water. Although tyrosine, it has predominantly non-polar structure, but hydroxyl group is there. So tyrosine is, some books will classify tyrosine into hydrophobic category, but it has got that hydroxyl group which can be polar. Okay, so that's why I put it into polar uncharged amino group. Asparagine has got this side chain amide nitrogen there, and also glutamine also has got side chain amide nitrogen. So like this, we have glycine, serine, threonine, cysteine, tyrosine, asparagine, glutamine. These are all polar but uncharged amino acids under physiological pH. Coming to negatively charged amino acids and positively charged amino acids. So let's look at positively charged amino acids first. So there are 3 amino acids that are positively charged. The first one is lysine, other is arginine, and the third one is histidine. Now lysine, the side chain of lysine, look at the side chain of lysine. This is CH2, 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 4 times CH2, and then we have NH3 plus in the side chain. So this is referred as butyl ammonium group. So butyl, that is CH2, CH2, so 4 times, that is butyl and ammonium, butyl ammonium group. That's what is there in the lysine. Look at arginine. Arginine has CH2, 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 and then it has got this guanidinium group. Guanidinium. So arginine has guanidinium groups. This is the guanidinium group, that is NH, C double bond NH and NH3 plus. So there is a positive charge there. Okay. So 3 nitrogens are there in the arginine, guanidinium. Remember that. That's important. You got to remember arginine has guanidinium group, and this is the one later it will, whenever it is participating in urea cycle. So the urea nitrogen that comes from here. So remember that. And then histidine. Histidine has got this imidazole group. Imidazole, and look at this imidazole group, it has NH plus there. So if you carefully look at the side chain of these amino acids, they have got positively charged amino group there in the side chain, and that's second positive charge. So because primary amino group, in each one of them primary amino group is there which has a positive charge, side chain also has got a positive charge. That means 2 positive and 1 negative. Primary carboxyl group is negative. So 2 positive and 1 negative. That means net positive charge is there in these 3 amino acids. Lysine, arginine and histidine. These are positively charged amino acids under physiological pH. When you compare 3 amino acids with one another, lysine and arginine are strongly positive amino acids, whereas histidine is a weakly positive amino acid. That's because the pK value of the side chain of histidine, that is imidazole group of histidine, pKa, whenever it is part of a polypeptide chain, it becomes around 7.0, which is equal almost nearer to physiological pH 7.4. That means 50% of this imidazole group will be protonated approximately, and approximately 50% will be deprotonated. So it has almost protonation and deprotonation. So it exists as weakly basic amino acids. Now let's look at negatively charged amino acids. We have 2 negatively charged amino acids. One is aspartate, which has got side chain CH2CO minus there. So that's negative charge is there in the side chain. And the other is glutamate. We have CH2CH2CO minus. That is, this is an alpha carbon alpha, and then the beta and the gamma. Gamma carbon is attached with the carboxyl group in glutamate. So there is 2 carboxyl group and 1 positive charge there. So 2 negative in each one of them and 1 positive charges in each. So net charge is negative here. That's why these are called as negatively charged amino acids, or it is acidic amino acid. Okay, negatively charged amino acid or acidic amino acid. Now let's look at classification of amino acid based on the polarity of the variable side chain as the overview of it. Whatever I have explained so far, I am basically reviewing them and giving you the list. Okay, let's look into the list again. So non-polar amino acids are alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, methionine and proline, and I have told you the mnemonic, it is PIM, POL, TV, PIM, POL, TV. Now polar but uncharged R groups containing amino acids, we have serine, threonine and tyrosine. These are 3 hydroxyl containing, all 3 of them contains hydroxyl group. And then we have glycine, asparagine, glutamine and cysteine. Note that cysteine has got thiol group, sulfur. Okay. Now polar amino acids but negatively charged under physiological pH are aspartic acid and glutamic acid, whereas polar amino acids which contain positive charge under physiological pH are lysine, arginine and histidine. Out of these 3, histidine is weakly basic amino acid, whereas lysine and arginine are strongly basic amino acid under physiological pH. Now let's look at a classification of amino acids based on the side chain characteristics, especially what is the structural arrangement of the side chain. By going with that, so we can classify amino acids as aliphatic amino acids, aromatic amino acids and heterocyclic amino acid and an amino acid. Okay, now let's look at aliphatic amino acid. So the aliphatic amino acids are basically they have got aliphatic side chain. That means they don't have aromatic ring or heterocyclic ring or a special amino group or something like that. Okay, so among aliphatic amino acids we have branched chain amino acids. The side chains of these amino acids are branched in nature. So those are valine, leucine, isoleucine. Now let me show you the structure of valine, leucine, isoleucine. So take a look at the side chain of valine, which has got CH and it is branched out as 2 methyl group there, and leucine again there is a branching there, and isoleucine there is branching. That's why these are, it has got aliphatic side chain because this is an open structure, no ring, and it is branched. That's why these are branched chain amino acid. Similarly, we have a sulfur containing amino acid. Sulfur containing amino acid. There are two sulfur containing amino acid. One is a methionine, where it has a thioether linkage. Sulfur is there, but it is not free. And cysteine. Cysteine has got thiol group, CSH2. So CH2, SH2, that is attached. So these are two sulfur containing amino acids. We have seen the structure of cysteine before. Now, amide group nitrogen containing amino acids, we have two of them, asparagine and glutamine. Because as both asparagine and glutamine, it has got amide group in the side chain. Let me show you that. You look at the asparagine amino acid, asparagine here, and glutamine. Look at the amide nitrogen, NH2 here. These are amide groups. That's why asparagine and glutamine, these are amide group containing amino acids. And then look at hydroxyl amino acids, hydroxyl group containing amino acids. We have three of them, serine, threonine and tyrosine. All three of them, they contain hydroxyl group. Previously, I have marked it, circled it. All three of them contain hydroxyl group. Now, simple amino acids. Simple amino acids are glycine and alanine. Because the side chain of glycine is just H. It is the most simplest amino acid out of 20 standard amino acids. Alanine side chain is just CH3. Now, coming to aromatic amino acids. Aromatic amino acids, they contain aromatic ring. So, there are three of them. One is tyrosine. Tyrosine is an aromatic amino acid. And then we have phenylalanine. Phenylalanine. And some books will mention tryptophan. Tryptophan, although it has got heterocyclic ring, but it has aromatic there. So, tryptophan. So, phenylalanine, tyrosine and tryptophan, aromatic. Now, amino acid. Amino acid is, it is proline. Amino acid that is special here is proline, because there is no primary amino group in this side chain of proline. It is interacted with the amino group. Now, coming to heterocyclic amino acid. Heterocyclic amino acid, they are histidine and tryptophan. Histidine and tryptophan are referred as heterocyclic amino acid. Now, acidic amino acids, which are also referred as dicarboxylic acids or negatively charged amino acids, are aspartate and glutamate. And the dibasic or positively charged amino acids or basic amino acids are lysine, arginine and histidine. Okay. So, these are all different classification of amino acids that you must be aware of. Coming to classification of amino acids based on their metabolic fate. Now, what is the metabolic fate of amino acids? Whenever amino acids are undergoing metabolism, especially when they undergo catabolism, so what is the fate of the carbon skeleton of amino acid? Because amino acid has got a carbon skeleton, it has got amino group. So, when you take out amino group and look at the carbon skeleton, what happens to the carbon skeleton in our metabolism? So, basically they will either form glucose or they will form ketone bodies. Some of them will form both glucose and ketone body. That's what is the metabolic fate of amino acids. So, some of the amino acids that form glucose, they are referred as glucogenic amino acid. And the amino acids carbon skeleton, if it forms a ketone body is like we have acetoacetate or beta hydroxybutyrate, these are ketone bodies. Those are referred as ketogenic amino acid. And some amino acids will make both glucose and ketone body, that is referred as both glucogenic and ketogenic amino acids. If you look at the table here, so each one of the category is written. So, glycogenic amino acid or the glucogenic amino acid, it is said in two ways. Either you call it as glucogenic or glycogenic, it is all same. So, here is the list of glycogenic amino acid. Let us not look into that right now. Let us look at the ketogenic amino acids first. So, the ketogenic amino acids, we have two of them, leucine and lysine. Okay, most of the books will write leucine and lysine as ketogenic, but in that the purely ketogenic amino acid is leucine, partly ketogenic amino acid is lysine. That's why we will just put it as leucine and lysine as ketogenic here. Okay, now look at both glycogenic and ketogenic amino acid. So both glycogenic and ketogenic amino acid, we have isoleucine, phenylalanine, tryptophan, tyrosine. The lysine here, some books will put that lysine into both glucogenic and ketogenic, but I would say it is more of a ketogenic amino acid than a glucogenic amino acid. It's predominantly ketogenic than glucogenic. So I'll just put it this side. So I just want to let you know that there may be some kind of controversy here. So you got to be aware of. So if there is you will have to go according to the question. Okay. If the question is about ketogenic and if leucine is not given but lysine is there in the choice, go for lysine. That's an answer. Okay. So if leucine is there, lysine is there and other amino acids are given, but you will have to choose one ketogenic amino acid in the choice, one best choice. At that time you just go for leucine even if lysine is present. So just go by the question. Anyway, so what are all the amino acids which are both glucogenic and ketogenic? It is isoleucine, phenylalanine, tryptophan, tyrosine, and some books will also put threonine into this list, both glucogenic and ketogenic. As it is there in the table here, it is glucogenic, it's placed into glucogenic predominantly, but it can also make some ketone bodies. So we have isoleucine, phenylalanine, tryptophan, tyrosine and threonine can be part of this group. So like this, so how do you remember glycogenic amino acid now? So once you know what are the ketogenic amino acids and both glucogenic and ketogenic amino acids, so if you remember them, you don't need to worry about remembering glycogenic amino acids. So what are the ketogenic? Leucine and lysine. I'll put this here. Leucine and lysine, both are ketogenic amino acid, and both glucogenic and ketogenic are isoleucine, phenylalanine, tryptophan, tyrosine and threonine. So once you remember them, so all other amino acids are glucogenic. So that's how you can remember. Now let's look at classification of amino acids based on the nutritional requirement. Before I explain you the nutritional requirement of amino acid, let me explain you what is nitrogen balance. So the nitrogen balance, it is the amount of nitrogen that gets into your body compared to the amount of nitrogen that is going out of our body. So nitrogen intake should be equal to the nitrogen output or excreted. So if the amount of nitrogen that is coming into our body, what is the predominant nitrogen that nitrogen that gets into our body? It is the amino acids. That's a protein that we all consume. You know, protein is amino acids are building blocks of protein, and each amino acid has that one nitrogen amino group which carry nitrogen there. At least one amino group is present in each amino acid. So that's how nitrogen gets into our body. So that is, I will write it as amino acids. Nitrogen gets in the form of amino acid. How nitrogen is excreted? What is the predominant excretory form of nitrogen in our body? It is the urea in the urine. Urea is the nitrogen excretory product because urea is NH2CO NH2. So amount of nitrogen that we are taking in should be equal to the amount of nitrogen that we are excreting, and that is what is referred as nitrogen balance, and generally adults are in nitrogen balance. Okay, now let's look at what is a negative nitrogen balance. Negative nitrogen balance is amount of nitrogen that is taken in is less than the amount of nitrogen that is excreted. That means proteins that are taken into our body is much less than the urea that is excreted. That means muscle proteins are breaking down here, because protein, major source storage form of protein in our body, majority of proteins in terms of quantity, they are present in skeletal muscle. So whenever skeletal muscle is undergoing kind of breakdown process, so there will be loss of nitrogen out of our body in the form of urea. So that means nitrogen coming into the body is less than the nitrogen that is going out of our body. That is what is referred as negative nitrogen balance. So example for that is starvation, when the person is not eating, or when person is in chronic disorder, suffering from chronic disorders, or if they are having any issues going on for a long period of time. So that can also put somebody under negative nitrogen balance, because muscle proteins are undergoing degradation in chronic diseases. So starvation, chronic diseases, any stressful events, road traffic accident where muscle injuries happened, that type of things will put somebody under. And even absence of essential amino acids that has to be taken in our body, that may lead to negative nitrogen balance condition. Coming to positive nitrogen balance. Positive nitrogen balance is amount of nitrogen that is taken in, amount of nitrogen intake is more than the amount of nitrogen that is excreted. That means urea excretion is much less. That means most of the nitrogen is retained in our body. So that means more and more proteins are made in the body. So that is what is referred as positive nitrogen balance condition. Example, pregnancy. During pregnancy, because fetus is growing inside, more and more proteins are made. And bodybuilding process or recovery from illness, because when the person is suffering from chronic diseases and the person is recovering, so there new proteins are made, so that means less urea is excreted. So recovery from illness, bodybuilding process, pregnancy, growth and development, like childhood where growth and development is going on, that's a positive nitrogen balance condition. So that's the background that I have given you to explain what is the nutritional classification of amino acid. So here is the nutritional classification of amino acid. We have essential amino acids, semi-essential amino acids and non-essential amino acids. Okay, so essential amino acids are the ones that are absolutely necessary for our body and it should be taken in the diet because we cannot synthesize them in our body. So essential amino acids, we cannot synthesize them because simply we don't have any enzymes to synthesize essential amino acids. We need to take them in our diet. So that you can remember essential amino acids as TV till PM. TV till PM. What is TV till PM? TV till PM is, it is tryptophan. T for tryptophan, V for valine, another T is, it is threonine, I for isoleucine, L for leucine, another L for lysine, P for phenylalanine and M for methionine. So this is what is TV till PM as a mnemonic to remember essential amino acids. Now what are semi-essential amino acids? Semi-essential amino acids are the ones that are needed in less quantities because some of the quantity of these semi-essential amino acids are synthesized in our body. Okay, like example for semi-essential amino acids, we have two of them, arginine, histidine. Arginine and histidine. Both arginine and histidine are referred as semi-essential amino acid. Arginine is the most semi-essential amino acid and histidine is partly, it is also referred as semi-essential amino acid. So when the question is asked, if only one choice need to be chosen, if both arginine and histidine both are there in the question, in the choices, you got to go for arginine, and then if arginine is not available, then your choice should be histidine. So when it comes to semi-essential amino acid, it is the arginine first. And then if it is not available, go for histidine. Okay. So arginine and histidine, they are synthesized to a limited quantities when there is a demand for more of them, like during positive nitrogen balance condition, like pregnancy, childhood, recovery from illness, bodybuilding process. At that time, we may need to supplement more arginine and histidine from the diet. That's what it means. Non-essential amino acids. Non-essential amino acids are the ones that are synthesized in our body, so you don't need to take them in the diet. So they don't need to be absolute component of our diet. Remember that. So this is what is a nutritional classification of amino acids. Coming to some special amino acids. So we have selenocysteine, which is referred as 21st amino acid, and then pyrrolysine, which is referred as 22nd amino acid. So 21st amino acid is selenocysteine and 22nd amino acid is pyrrolysine. Let's look into little bit more on selenocysteine and pyrrolysine. So selenocysteine, it occurs in the active site of several enzymes in our body. What are the enzymes that contain selenocysteine, which is a 21st amino acid? And that is, we have thioredoxin reductase enzyme, which is involved in nucleotide, deoxynucleotide biosynthesis. And then we have glutathione peroxidase. Glutathione peroxidase, it acts as a scavenger of peroxides. Basically, hydrogen peroxide related reactive oxygen species or free radicals can be scavenged by glutathione peroxidase mediated action, which is a selenocysteine containing protein. And then we have deiodinase enzyme involved in T3, T4, thyroid hormone synthesis, and then we have glycine reductase enzyme in the glycine metabolism, and then selenoprotein P. So these are selenocysteine containing proteins. So that is thioredoxin reductase, glutathione peroxidase enzyme, and then we have deiodinase, glycine reductase, and selenoprotein P. Let's look briefly into how exactly selenocysteine become part of our polypeptide chain, because selenocysteine do not have a standard codon for selenocysteine. So we have three stop codon, UGA, UAG, and UAA. Now out of these three stop codons, which don't code for any amino acid, basically it stops the translation process, but the speciality here is UGA, which is a stop codon normally. So this UGA, it will undergo something called as translational recoding process. Okay, translational recoding. So what happens in this translational recoding process is, so whenever UGA comes in, at that time this UGA, especially with the sequence here, selenocysteine insertion sequence, that is SEC-IS, selenocysteine insertion sequence. So whenever that selenocysteine insertion sequence, which appears on the mRNA with the UGA codon appearing, so selenocysteine machinery, basically selenocysteine synthase enzyme coming in, and what it does, it is going to code that selenocysteine with the tRNA, which is specially coming for serine. It is going to convert that tRNA, which is meant for serine, into tRNA for selenocysteine. So that's how the serine is converted into cysteine, selenium atom is there, and that's how selenocysteine is basically incorporated into selenoprotein molecule. So make sure you remember, stop codon UGA will undergo translational recoding, so that it is coding for selenocysteine in all selenocysteine containing proteins called selenoprotein. I have given you the list of them. So that's the basic information that you must remember. Coming to pyrrolysine. Pyrrolysine is a 22nd amino acid which is encoded by another stop codon, that is UAG. Make sure you remember that. So for selenocysteine it is UGA. Don't forget that. That's the stop codon UGA, which is undergoing translational recoding to make selenoproteins. Okay. Whereas pyrrolysine, it is coded by another stop codon UAG. But only difference here is, pyrrolysine we don't see them in mammalian proteins, usually they are found in prokaryotes. So what happens here is, so this pyrrolysine, it is similar to lysine, but pyrroline ring is attached to the side chain of lysine to make pyrrolysine using UAG codon. So that's in the prokaryotes, remember that. Now let's briefly go into what is the post-translational modification of some of the amino acids, especially when they are part of a polypeptide chain. Whenever they are attached to a polypeptide chain, they can undergo post-translational modification process. What are the post-translational modification of these amino acids? One is hydroxylation, where hydroxyl group is attached. Glycosylation, carbohydrate is attached to a protein molecule. Lipidation, lipid is attached. Methylation, methyl group is attached. Acetylation, acetyl group is attached. Carboxylation, carbon dioxide attached. Phosphorylation, phosphate, PO4 minus attached. So all these processes can happen whenever a protein amino acid is part of a polypeptide chain, and post-translationally or co-translationally these modification can go on. So example, each one of them I have given you the figure pictures in the form of figure. So we have glycosylation, carbohydrate addition here. Usually carbohydrate is attached to the hydroxyl group of serine, threonine, tyrosine, because these are hydroxyl containing amino acid. That type of carbohydrate attachment to the hydroxyl group of serine, threonine, tyrosine, that is referred as O-glycosylation process, O linkage. Here is the O linkage. Hydroxyl group is attached with the carbohydrate. Now, if a carbohydrate is attached to the amino group of asparagine, asparagine, it's a polar but neutral amino acid. So, amino group of asparagine, if the carbohydrate is attached, that type of attachment we call it as N-glycosylation. Here is the asparagine, with amide nitrogen that is attached with the carbohydrate. So, it is an N-glycosylation. So, in a protein molecule, whenever it is undergoing carboxylation process, so we have O linkage and N linkage, O glycosylation and N glycosylation. Out of these two, the most common type of glycosylation is N glycosylation, remember that, very important. And asparagine is the amino acid that accept the carbohydrate group. Coming to lipidation, attachment of lipids like palmitic acid is a fatty acid. So, which is referred as palmitylation, attachment of palmitic acid. Myristicylation, myristic acid being attached to. So, palmityl group, palmitic acid attached to the cysteine thiol group. So, cysteine has got thiol group SH, to that palmitic acid is attached. Whereas myristicylation, it is attached to the amide, the amino group of glycine. You know, glycine as a primary amino group, and to that it is the fatty acid is attached, myristic acid is attached. Prenylation, prenyl group attached to the cysteine, thiol group of cysteine, prenylation process going on. So, palmitylation, myristicylation, prenylation, these are the lipidation, post-translational modification of amino acids. Coming to phosphorylation, one of the most common post-translational modification I would say is phosphorylation, which is reversible. So, where phosphate is usually attached to serine, threonine and tyrosine, hydroxyl group of serine, threonine and tyrosine, usually serine and threonine. So, undergoing phosphorylation process. So, acetylation, acetylation is acetyl group is usually attached to the butyl ammonium group of lysine. You know, lysine has got the CH2, 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 CH2 and NH3 plus, and that butyl ammonium is attached with acetyl group. That is what is referred as acetylation, remember. So, whenever acetylation is going on, it is the butyl ammonium ion of lysine which is attached with the acetyl group. Coming to carboxylation, carboxylation is carbon dioxide is attached to a amino acid. Example, we have glutamate. You know, glutamate has it has alpha carbon and beta and gamma. Gamma carbon, usually generally it has got a carboxyl group attached. Normally, that's what is glutamate. One more carboxyl group is attached to the gamma carbon. That is what is gamma carboxylation. Generally, gamma carboxylation of clotting factor 2, 7, 9 and 10. Clotting factor 2, 7, 9, 10, protein C, protein S, matrix GLA protein, osteocalcin, all of them will undergo gamma carboxylation of glutamate residue. Okay, that's an example for carboxylation process. And then hydroxylation. So, like proline and lysine, amino acid proline and lysine, they can be attached with hydroxyl group. Usually this happens during collagen biosynthesis. You are going to study that in collagen biosynthesis. Hydroxyl. So, proline and lysine converted into hydroxyproline, hydroxylysine by prolyl hydroxylase, lysyl hydroxylase enzymes. Okay, that's the post-translational modification of proline and lysine. Coming to some of the amino acids that can be of D form, which may come into our body either in the form of drugs or they may be coming from the bacteria. So, those are D-amino acids, because predominantly most of our amino acids in our body, they are of L form, LEVO form. You can find some of the amino acids in our body as a D form. So, like D-glutamic acid, D-alanine. Example, like we have valinomycin, gramicidin A, gramicidin S, actinomycin D. These are all the drugs which are synthetic ones and used in the treatment. So, those are of D forms of amino acids. Like then we have D-aspartate and D-serine, D-methionine, D-tyrosine, D-leucine, D-tryptophan. They are all coming from the bacteria. Okay. So, these are bacterial products. So, they can get into our body. Now, coming to some of the special properties of amino acids. One of the special property of amino acid is absorption of ultraviolet light by proteins. So, whenever proteins are exposed to ultraviolet light, so they will show a characteristic absorption band which is around 280 nanometer. So, the 280 nanometer absorption pattern of proteins are usually contributed by aromatic amino acids. So, we have three aromatic amino acids, tyrosine, phenylalanine and tryptophan. So if you observe this particular figure carefully, look at the tryptophan here. Now the absorption band of the tryptophan is high compared to tyrosine and phenylalanine. That's why the amount of light that is absorbed by the tryptophan is most by out of all three aromatic amino acids. Tryptophan absorbs most of that. That's what I have written in the first statement. Aromatic amino acids, specifically tryptophan, contributes to the ability of most proteins to absorb light at 280 nanometer range. Now, this particular property of absorption of light at 280 nanometers, especially by tryptophan, it is used as an indirect measurement of protein concentration in a solution using ultraviolet light. Remember. So, remember about the role of tryptophan in absorption of ultraviolet light at 280 nanometer. Coming to briefly buffering action of amino acids in a protein molecule, and we will take example of histidine. Histidine exerts a maximal buffering capacity in a protein molecule because the side chain of histidine, imidazole group, whenever it is in individual form, it is 6. Whenever it is part of a polypeptide chain, it moves to 7. Okay, so that means now the pK of imidazole group is 7.0, whereas the normal pH is 7.4. Now it is falling something like this. pH is almost equal to pK. That means 50% is protonation, 50% deprotonation. And also you need to remember that the maximum buffering capacity of a group, it will exist between pKa plus or minus 1 in that range, plus or minus 1 pH. pKa plus or minus 1 pH range, that's where the maximum buffering capacity will be there. Let me show you that here. Look at R group, pKa is 6. Now here is the 6, and now 6 pKa 6 plus or minus 1 pH. That means between pH 7 and 5 and 7. So because it is 6 in the center, plus 1 and minus 1, right? Plus 1 pH, minus 1 pH. Okay. So that means pH 5 to pH 7. So between pH 5 to 7, it has got that maximum buffering capacity. That's why you see this deflection in the line there, right? It starts only after this range. That is, this will be 5 and this will be 7. Only after that there will be deflection. Okay. Similarly, you can observe that in the primary carboxyl group, primary amino group also. For primary carboxyl group, pK is 1.8. Now note that pKa plus or minus 1 pH, that is 0.8 to 2.8, in that range. There is no deflection. That's a buffering area. Similarly, for amino group 9.3, so 9.3 minus 1 plus 1. Okay. So in that, this is where the buffering area is there. So because at that time it exists as equal number of positive, equal number of negative charge, so there is no change in the pH there. So that's what is the buffering capacity, which we can understand using histidine, because histidine is as it has got, as I said before, whenever it is part of a polypeptide chain, pK and pH is almost near, so 50% protonation, 50% deprotonation. Coming to certain plants which contain L amino acids that can adversely impact our human health when they are consumed. Okay. So whenever these plants are consumed, because they contain some neurochemicals that can affect our neuronal system, it can lead to neurodegeneration, neuronal issues, neuronal conditions. So I am just going to give you the names of these toxins, like a disease called neuroletharism, where there will be L-homoarginine and beta-N-oxalyl alpha-beta diamino propionic acid, which will lead to neuroletharism. And then we have osteoletharism here, osteoletharogen, which is a beta-N-glutamyl aminopropionitrile. And then 2,4-diamino butyric acid that inhibits hepatic urea cycle and ornithine transcarbamylase enzyme in the urea cycle, leading to hyperammonemia and leading to neurological issues. And then we have beta-methylaminoalanine, which is a risk factor for development of amyotrophic lateral sclerosis, Parkinson dementia complex. So these are the names of some of the chemicals that can lead to certain neurological issues. And lastly, I am going to give you what happens to amino acids when they undergo decarboxylation of their primary carboxyl group. So here is the alpha carbon, COO minus, NH3 plus, and then H, and the side chain. So side chain I will write for tyrosine, it is a benzene ring with the hydroxyl group. Okay. Now, if I remove carboxyl group of this amino acid, so here is the carboxyl group here. If I remove that, so basically what will be remaining is only this part here. It has got amino group, primary amino group is remaining, carboxyl group is taken out. CCO, it will be released as carbon dioxide. Okay. C double bond O. So it's a decarboxylation of tyrosine. So when I am doing decarboxylation of tyrosine, primary carboxyl is removed as carbon dioxide. Tyrosine is now becoming tyramine. It's a corresponding amine. So decarboxylation of amino acid will become a corresponding amine. So I have written corresponding amines here. So histidine will become histamine. It will participate in inflammatory process. Tyrosine become tyramine. Tryptophan become tryptamine, lysine become cadaverine, glutamic acid becomes gamma amino butyric acid, which is a inhibitory neurotransmitter, serine becomes ethanolamine, which is used in membrane phospholipid formation, cysteine become beta mercaptoethanolamine. Okay. So these are the different corresponding amines that you can get from decarboxylation of amino acids, which will participate in certain reactions, inflammatory process, biological reactions. Some of them act as a neurotransmitter, some of them are used in phospholipid membrane formation. So that's about amino acids lecture. Thank you.